Dr. Leena Meshram. I'm working in Mahatma Phule College as an assistant professor in zoology. Today in this video series lecture, we will talk about immunoglobulins. We'll talk about immunoglobulins. Now, immunoglobulins are also called as antibodies. Now, in the previous lecture, I have told you about antigens. Antigens are antibody generators. Antigens are antibody generators. So, these immunoglobulins, they are also called as antibodies. They are immunologically active serum globulins, that is, they are proteins. Now, when these proteins are produced, they are produced when an antigen enters into the body. It evokes an immune response. Basically, it is called as humoral response, where the B cells will convert into the plasma cell and produce these antibodies. Okay, so these immunoglobulins, they are glycoproteins and they are produced by the plasma cells in response to an antigen. Very important, when an antigen enters into the body, it will trigger the immune system, and then the B cell will get modified to plasma cell to produce this antibodies. So when antibodies are produced, these antibodies will neutralize the reaction of antigen. Antigen binds with antibody and will neutralize the reaction of antigen. So when an antibody reacts with a specific antigen, it is called as antibody. So when an immunoglobulin, it reacts with specific antigen, it is called as antibody. Okay, when I told you that all the B cells produces immunoglobulin, all the immunoglobulins are antibodies. See, when immunoglobulin does not show specific antigen specificity, it is called as immunoglobulin. So B cells will produce immunoglobulins. These immunoglobulins, when they combine with antigen, then it is called as antibody. So all antibodies are immunoglobulins, but all immunoglobulins are not antibody. Since immunoglobulins are produced by the B cells, they are produced, for example, IgM. It is present on the B cell. It is the most heavier immunoglobulin which reacts with the antigen, and it is present on the B cell. Immunoglobulins are 5 distinct classes: IgG, IgA, IgM, IgD and IgA. There are 5 classes of immunoglobulin, and each class differ from the other on the molecular weight, amino acid, the carbon content and the biological property. Okay, so what did I tell you is immunoglobulins are produced by the plasma cells. They are produced by the plasma cells. These immunoglobulins, when they react or when they bind with the antigen, then it is called as antibodies. And all antibodies are immunoglobulin, but all immunoglobulin may not be antigen. This is the structure of immunoglobulin. You can say that it is a Y-shaped structure. It is a Y-shaped structure. So when you say, see, IgG is the abundant immunoglobulin present in the blood. Okay, now this is the basic structure of immunoglobulin, which shows you that there are two heavy chains and there are two lighter chains. It is a Y-shaped structure. It is a Y-shaped structure showing two heavy chain and two lighter chain. Okay. Now this force polypeptide structure. It is made up of 4 polypeptide structure. This is a heavy chain, this is the heavy chain, this is the lighter chain, this is the lighter chain. All these 4 polypeptide chains, they are held together by disulfide bridges in between. They are held together by disulfide bridges in between. It will have C terminal and N terminal. This is the heavy chain having the N terminal and having the C terminal, which is also called as carboxylic end. This is an N terminal, this is an C terminal. So each heavy chain and each lighter chain will have the C terminal and N terminal. Now this N terminal is called as an variable end and this terminal is called as a constant end. It is called as the constant end. Okay. So when you see the structure was given by Porter. It was given by Porter. The structure suggests that it has 4 polypeptide chain. It has 4 polypeptide chain with a Y-shaped structure. As I told you, it has 4 polypeptide chain. Each is called as a single polypeptide chain having an C terminal and N terminal. So the immunoglobulin, it is made up of 4 polypeptide chain, 2 heavy chains and 2 lighter chains. Light chain is denoted as L, heavy chain is denoted as L. So 2 identical L chain and 2 identical light chain. Each terminal will have an N terminal and it will have a C terminal. The 4 polypeptide chains are connected by disulfide bonds and the other non-covalent bond. So you can see here, this is the heavy chain, this is the lighter chain. The heavy chain is connected to the lighter chain by the disulfide bond. With the disulfide bond, each chain will have a heavy chain and a lighter chain. The heavy chain is connected to the lighter chain with a disulfide bond having a variable N and a constant region. Okay, can see the structure very nicely. So this is the variable domain, this is the variable domain and this is the constant domain. It is the constant domain. Now when you come to the lighter chain, lighter chains are 2 types, and both are identical. There are 2 types and both are, and in one immunoglobulin, both the light chains will be same. Okay, they are either kappa type or they are either lambda type. So they are K type or they are lambda type. When both the light chains are kappa type, it is called K type, and when both the light chains are of lambda type, they are called as L type. Okay, so each light chain will have approximately 214 amino acids. So when we say it is a Y-shaped structure having light chain and heavy chain, the light chain will have approximately 214 amino acids with the molecular weight of 25 daltons. When you talk about heavy chain, the heavy chains are distinct for each 5 types of or 5 classes of immunoglobulin. Previously when I told you that there are 5 classes of immunoglobulin, that is IgA, IgG, IgD, IgM and IgT. each one will have its unique type of heavy chain. So IgA will have alpha heavy chain, IgG will have gamma heavy chain, IgD will have delta heavy chain, IgM will have mu heavy chain, and IgE will have epsilon heavy chain. The heavy chain has approximately 428 amino acid. Okay, so the heavy chain will have 428 amino acid long. The polypeptide chain is almost 428 amino acid long. This four chain, they are connected to each other by the disulfide bridges and by the non-covalent bond like hydrophobic bond, ionic bond, van der Waals forces of attraction. So all these four polypeptide chain, they will form a three-dimensional structure, and they are interconnected to one another by the disulfide bond and non-covalent bond. So interchain there is present between the heavy chain and light chain and heavy chain, and intrachain which is present between the light or the heavy chain. So these bonds, the non-covalent bonds and the disulfide bond is present between the two heavy chain and one between the heavy chain and the light chain. The heavy chains are linked with one another by disulfide bond. The number may be from 1 to 13. Each light chain is also linked with the heavy chain by an inter-disulfide bond. Each light chain has two intra-chain disulfide bond, while the heavy chain will have 4 intra-chain disulfide bond. The heavy chain are held by 2 interchain disulfide bonds. Okay. Now the variable region and the constant region. The light chains are divided into variable and constant region. As I told you, there is a variable domain and there is a constant domain. The light chain consists of one variable domain. The heavy chain also consists of one variable domain, and in case of IgG and IgA, they will have 3 constant domain. The variable regions are responsible for antibody binding, and the constant regions are responsible for various biological functions like complement activation and binding of the cell. Coming to the structure, see, this is the structure of showing the heavy chain and light chain. This is immunoglobulin G, where it will have three constant domain and one variable domain. Okay, so these constant the domains are formed because of the heavy foldings of the polypeptide chain, and they are connected by the disulfide bridges. As we have studied earlier, there can be interbond and intrabond of the disulfide bridges. Okay, so interbond between the two different heavy chain and the light chain, and interbond between the same heavy chain and the light chain. Okay, so what you can see in this is the immunoglobulin G will have one domain which is a variable domain and 3 domain which will be the constant domain. This variable domain of an immunoglobulin is responsible for binding with the epitope of an enzyme and of an antigen, and hence it is called as paratope. This is a variable region which can bind with the antigen. So this is called as paratope, which binds with the epitope of an enzyme. Okay, so I hope the structure of immunoglobulin is clear to you. It is very simple. When it is asked in the exam, the basic structure of immunoglobulin, what you have to say, what you have to write about is, again I will tell you in just 1 minute, that it is made up of 4 polypeptide chain, and immunoglobulin, it is made up of 4 polypeptide chain. There are two heavy chains and two lighter chain. Two heavy chain, these are heavy chain, and these two are lighter chain. Okay, the two heavy chains are linked by each other with a disulfide bond, and one light and one heavy chain is also linked by the disulfide bond. There are also other non-covalent bonds which will keep this structure together, and that is hydrogen bond, ionic bond, and there are hydrophobic bonds also which will keep the structure. Then you can see that there is every chain will have an N terminal and a C terminal as it is a polypeptide chain. The N terminal is a variable region, and where it is C terminal, it is a constant region. The N terminal is responsible for binding of antigen antibody. Okay, so I think I'll stop here. I hope you understood the structure of basic structure of immunoglobulin.